Good day, everyone. My name is Idowu Adeosun, a PhD candidate at the Department of Biochemistry, Genetics, and Microbiology at the University of Pretoria, South Africa. First, I would like to thank the Beyond Science Initiative for availing me this opportunity to present part of my research findings at this year's conference. I'll be presenting on the topic molecular modeling of SdiA protein by selected flavonoids and terpene compounds to attenuate virulence in Klebsiella pneumoniae. Here's an overview of my presentation. I'll be taking you through the introduction, the methodology employed, the results obtained, the conclusion, and the acknowledgements. Klebsiella pneumoniae is a gram-negative bacteria that belongs to the family Enterobacteriaceae. It has been characterized as one of the worrying and escape pathogens owing to its ability to escape virtually all classes of antibiotics, including the last-line resort of antibiotics known as the carbapenems. According to the World Health Organization, Klebsiella pneumoniae has been categorized under the priority 1 critical pathogens, that is the group of organisms for which new treatment options are urgently needed. This pathogen has been reported to account for about 1/3 of all gram-negative infections, such as meningitis, pneumonia, bacteremia, liver abscesses, among others, resulting in increased morbidity, mortality, and extended rate of hospitalization. Klebsiella pneumonia's virulence can be linked to quorum sensing, which is a bacterial cell-to-cell communication that uses signaling molecules or chemical molecules known as autoinducers in a high bacterial cell population density. According to Kosa et al. 2019, this mechanism does not just enhance the pathogenesis of those bacteria, but also improves their ability to severely infect and damage their hosts. In gram-negative bacteria, the transcriptional regulator LOX-R protein helps to detect the autoinducers, which are the AHLs, that are being produced by the LOX-I synthase. However, some Enterobacteriaceae, such as Klebsiella pneumoniae, do not possess this LOX-I synthase that helps to produce the AHL molecule. Instead, they encode the SdiA protein that helps to recognize the AHLs that are being produced by the mixed community genera. This SdiA, which is the LOX-R transcriptional regulator protein, is often linked to quorum sensing, cell division, and virulence factors in Klebsiella pneumoniae, which consequently marks SdiA as a potential therapeutic target in Klebsiella pneumoniae. Hence, impeding the signaling system that is often facilitated by this SdiA protein can hypothetically render this pathogen less or non-virulent, thereby posing novel therapeutic management of its infections. The question now is, how can we inhibit this quorum sensing associated SdiA protein in Klebsiella pneumoniae? According to Coe et al. 2013, medicinal plants and their secondary metabolites can be the next generation magic bullets, as they can mimic the microbial signal molecules to compete for the protein active site and also thwart the signaling mechanism, thereby hindering the expression of virulence factors. This study therefore employed molecular modeling approach to evaluate the antivirulence potential of plant secondary metabolites against the SdiA transcriptional regulator in Klebsiella pneumoniae. The strategy employed as follows. First, the sequences were retrieved from the Kyoto Encyclopedia of Gene and Genome database. The template was identified using the NCBI BLAST-P, and the homology modeling was carried out using the Swiss model server, where the 4LFU was used to model the template that was used, and this SdiA modeled protein was docked against 31 selected compounds using the Schrödinger software. After the molecular docking was completed, molecular dynamic simulations was also conducted to study the atomic and the interatomic interactions of the protein ligand system to validate the stability of a protein ligand complex, as well as to estimate the binding free energy of these compounds, which is indicative of therapeutic properties. The ADMET prediction was also done to validate their drug likeness properties. Here are the results obtained from this study. This table shows the docking result of the selected compounds against the SdiA receptor protein of Klebsiella pneumoniae. Of the flavonoids and terpenes tested, for the flavonoids, genistein was shown to reveal the highest docking score of -9.752 kilocalorie per mole, while for the terpenes, phytol was shown to reveal the highest docking score of -9.205 kilocalorie per mole. These scores are regarded as good docking scores as they range between the -5 and -15 kilocalorie per mole. And these scores are regarded as good. This implies that these compounds were able to fit perfectly into the pocket of the receptor protein. This figure shows the interaction network between the SdiA protein and the best docked compounds, which are the phytol glycine, in comparison with the fluoronosylborough diester, which is the AI2 molecule, which serves as a reference compound in the study. From this interaction network, it was shown that hydrogen bond was formed between the phytochemical compounds and at least one amino acid residue in the SdiA protein, both with phytol glycine as well as the fluoronosylborough diester. This hydrogen bond helps to strengthen the bond between the compounds and the SdiA receptor protein, which confers more specificity and directionality. This could have helped with the good docking scores that was obtained during the docking simulations. Molecular dynamic simulations was also conducted, and this helped to shed more light on the root mean standard deviation. And when this was studied, it helped to reveal the overall conformational stability of this protein when bound to the test compounds. And the scores obtained went from 1.48 Armstrong to 2.58 Armstrong for the terpenes, while for the flavonoids, the scores ranged from 1.44 Armstrong to 2.50 Armstrong. And this was done at 100 nanoseconds simulation production time. Similarly, the root mean standard fluctuation was also conducted, which helped to elucidate the flexibility and the motion of the individual residues in the protein, with the scores ranging from 1.08 Armstrong to 3.74 Armstrong for the studied compounds. This table shows the root mean standard deviation and the fluctuation of the best docked terpenes and flavonoids, where sabinine and genistein were shown to reveal the lowest RMSD scores of 1.48 Armstrong and 1.54 Armstrong respectively for the terpenes and flavonoids, and this corresponds to the compounds that enacted the most structural stability. Also, sabinine and quercetin reveal the least RMSF scores, and the lower this RMSF score, the higher the stability of these compounds. This figure shows the comparative RMSD plots, which reveals the degree of the stability and the convergence of the studied terpenes and flavonoids over the 100 nanoseconds simulations time, where sabinine was shown to reveal the least RMSD score for the terpenes. However, for the flavonoids, genistein was shown to reveal the least RMSD score. Similarly, this chart reveals the RMSF, that's the root mean standard fluctuation of each of the residues of the protein over the 100 nanosecond simulation production time for both the flavonoids and the terpenes when superposed on the crystal structure of the system, which helped to show the differences in the fluctuation as well as differences in the conformation or the conformational changes. The binding free energy landscape of the SdiA bonded terpene and the flavonoid compounds were then estimated. According to literature, molecular mechanism computation often combined with continuum summation model to derive binding free energies for macromolecules, and this approach represents a higher level scoring theory than molecular docking. Hence, binding free energies were estimated in the study. And the results showed that phytol had the highest binding free energy for the terpenes, while for the flavonoid, genistein was shown to reveal the highest binding energy of -68.1393 kilocalorie per mole. Binding free energy has been regarded as the single most important initial indicator for drug potency, and since these compounds were shown to reveal high binding free energy, which is indicative of their drug potency, they were further validated for drug likeness properties. This was done using the Lipinski rule of 5, which says that the molecular weight must be less than 500, the log P or the lipophilicity must be less than 5, the hydrogen bond donor must be less than 5, and the hydrogen bond acceptor must be less than 10. This table shows the conformation of these compounds to the Lipinski rule of 5, where all of the test compounds were shown to obey the Lipinski rule of 5. 8 of them showed no violation to all of the rules. However, 9 showed one violation each, which is still regarded as acceptable. Here is a table showing the other parameters that were obtained during the drug likeness validation of these phytochemical compounds, and these parameters include their physicochemical properties, their lipophilicity, water solubility, the pharmacokinetics, as well as the medicinal chemistry. The physicochemical properties have been discussed in the Lipinski rule of 5, which talks about the hydrogen bond donor, the hydrogen bond acceptor. The lipophilicity was also studied, and the values, the scores obtained are shown in this column. And that the lipophilicity is important, especially for the pharmaceutical industry, as this helps to shed more light on the potency of the drug and how they also have affinity for a lipophilic environment. The water solubility was also assessed, where all of these compounds were shown to be soluble in water, except for phytol, which revealed moderate solubility. And this solubility shown for those compounds further revealed them as compounds that can be easily absorbed and dissolved. For the pharmacokinetics, where the blood-brain barrier permeability was studied, most of these compounds were revealed to be able to permeate the blood-brain barrier, which means they can permeate the central nervous system, which is indicative of therapeutic ability. In medicinal chemistry, the paints and point structural alert often predict unstable or reactive or toxic fragments in the structure of these compounds. And from our results, all of these compounds were shown to reveal zero pain alert, which further reveals them as potential drug candidates. In conclusion, the study was able to validate the interaction between the SdiA autoinducer binding site and the studied compounds, forming critical interactions with the binding site key residues. And molecular dynamics simulations established the inhibitory performance as well as the interactions of these compounds, revealing their free binding energies that are as high as minus 68 kilocalorie per mole. The drug likeness prediction of these compounds validated their drug potentials, thereby conferring them as promising quorum sensing inhibitors for Klebsiella pneumoniae. Findings further suggest the exploration of these phytochemical compounds as alternative quorum sensing inhibitors and also examining their antivirulence potential against Klebsiella pneumoniae. Findings from this study have been published in the Journal of Biomolecular Structure and Dynamics, and the study was done in collaboration with the University of KwaZulu-Natal, Durban in South Africa, as well as the Ahmadu Bello University, Zaria in Nigeria. On this note, I would like to acknowledge my supervisor, Dr. Kosa, for a job well done on the supervision of this study. I also like to acknowledge my lab mates for their support, Prof. Zeno for assistance with the molecular docking software. I'd like to acknowledge the Center for High Performance Computing, CHPC at Cape Town, for providing a platform for the molecular docking and the molecular dynamic simulations. And lastly, I would like to acknowledge the University of Pretoria for the Commonwealth Scholarship. Thank you so much for listening.